My name is Howard Edelman. I'm your host for Israel and the Jewish World. We begin with our regular feature on the weekly news from Israel. Then we move to the third in our series on IMRC, the Institute for Medical Research, Israel, Canada at Hebrew University. We explore senescence, the study of the biological changes related to aging and the weakening of the body's mechanism for preventing cells reproducing anarchically, as they do in cancer. For as we age, we lose the preventive mechanisms that keep cells from growing wildly, as if they were in search of immortality, even at the expense of the body. In parallel, we interview 4 academic experts to explore why the overwhelming majority of Palestinian Israelis are law-abiding. What might make some rise in rebellion? Finally, we end with some reflection by our resident rabbi on the archetypal biblical rebel, Korach. Stay with us for an exploration of why some cells rise in rebellion. My guest is Ittai Ben-Porath. He's another guest in the series on innovation in science in Israel. Welcome to the show, Ittai.
Nice to be here.
Now, you're part of this complex of this Institute for Medical Research, Israel, Canada, and you are one department of a big complex of research. What's your department?
Correct. I'm a member of the Department of Developmental Biology and Cancer Research, which is part of the Institute of Medical Research under the Faculty of Medicine at the Hebrew University.
Now, you started at Hebrew University, your training. You didn't start as a medical student.
Correct.
You were a science student?
I was a biology undergrad at the Hebrew University, and then I continued on to do my PhD thesis in the university.
But it was in genetics, I believe.
Correct. So after the field of at that time, I belonged to the Department of Genetics, and the work was much focused on molecular biology and genetics.
But then you wanted to, how'd you get interested in the link between genetics? Because you could study genetics without studying diseases, but you got interested in the links between genetics and disease. How did that happen?
Right. So I think even the second year of undergrad, in a very basic course about cancer, biology of cancer, and genetics of cancer, I was really struck by the beauty of the basic discoveries in this field that really demonstrated that cancer is caused by genes that malfunction. And that caused me at that stage, led me at that stage to start working or doing some research in one of the labs.
As an undergraduate
As an undergrad.
They let undergraduates in?
Oh, they encourage them because that's the way, once they pull you in.
I see it.
I try to do this today myself.
I
right?
see.
Because if somebody is fascinated, from within, they will often seek to do this work at an early stage, and this is usually a great driving force in your work.
What was your thesis? What was your PhD thesis?
So at that time, we were interested in a gene that's very important in cancer. It's called MYC, M-Y-C. It's one of the very earliest genes that was associated with cancer at all in the late 70s. Its discovery is a seminal discovery in the field. But for a long time, people didn't understand what MYC does. In effect, it's a gene that turns on other genes. So on the one hand, you understand that it's kind of a programming gene. It has broad effects. It's doing something very major. But on the other hand, you remain with the question, well, what happens underneath it, downstream of it? Who does it activate, and which other guys are really carrying out those essential duties that MYC is trying to effect in the cells? And we were interested in finding such genes that are controlled by MYC and execute some of these cancer-related functions.
So who works for MYC? And did you document who did it and how the mechanics work?
So
it's like a structure of a labor crew. Here's the boss coming along telling us what to do.
Exactly. And it's actually very difficult to figure this out. I was working on one single gene that we were able to show that if if it's not there, then MYC has trouble making a cancer cell grow and divide and be aggressive. In later years, it turned out that actually MYC, it's very, very difficult to pinpoint MYC's actions in this type of way, because what it turned out, and this really came out only in the recent 3-4 years, is that MYC controls thousands of genes at once, and each one of them only to a quite small extent. So what you get is really kind of a concerted effort of thousands of genes controlled by this one master. So actually the attempt of our attempt and several other labs in parallel to find that one critical target, there was never one single important target. So it's a very important insight about how this type of gene like MYC really affects a cancer cell. It's not a simple chain of A affects B affects C in this case, but it's A doing a very subtle and broad range effect, and many people are still trying to figure this out.
Well, it's interesting, the very process you start with the false assumption, i.e. that it's after one, and after you do a lot of work, it's not. You're wrong. Right. But then you learned a lot more about the gene.
Absolutely.
Now you went off for 6 years to MIT in Weinberg's lab.
Correct.
What did you do under Weinberg? Because he's very famous in this field.
Right. So Weinberg, really, Bob, as we, I apologize, as we refer to him, what really characterizes his working mode and his laboratory is that every several years he kind of picks up his head and looks around and says, what's the most important question in cancer biology that exists today about which we know really nothing? And that way he was able over the years to make very important contributions. When I joined the lab, there were two main issues that we were thinking about, and I actually kind of touched upon both. One topic was the process of how the body normally blocks cancer development. If you have a normal cell, the body aims to keep this cell in check. The worst thing that can happen is that one cell decides to go wild, things go awry, it starts to divide on its own, ignores its environment. That what leads you to cancer. So the body has external and internal mechanisms to prevent this.
So it doesn't like anarchy.
The cell.
No, the body, the whole organism.
It doesn't like anarchy. Absolutely, that's the worst. We're comprised of billions and billions of cells, and if each one of them did what it wanted to do, ignoring the rest, we wouldn't be here conducting an interview. But it turns out that within cells, there are really mechanisms that keep the cell in check. One of them is termed cellular senescence. And in essence, if a cell starts dividing and dividing, proliferating, generating daughter cells, there's a clock within these cells that says, you know what, this is enough. There's no way this cell, you're allowed to divide so many times. There's a stop mechanism that at a certain point,
that's called senescence.
Yeah, because originally this was observed by growing cells in a dish in culture, in the incubator, and a very important scientist named Hayflick found that you can divide them and you can keep them dividing for only so long, and then they'll stop and senesce, age, and at that point they'll remain alive, but they won't divide anymore. And this is unlike cancer cells, which are immortal. They can grow forever and forever. They never stop. So obviously,
except until they kill their host.
Absolutely.
And then they stop.
Absolutely. So the cancer cell basically ignores this mechanism. So here's a major question: what is this mechanism? How does a normal cell know to stop, and the cancer cell?
And what was your contribution specifically?
So it turned out that within normal cells, there's a particular mechanism that triggers the senescence program. It's like a switch on. And that resides in a structure within the cell, which is on the chromosomes, those bodies that carry the genetic information, and each cell has 46 of those. And these are like long sticks, and they have ends. The ends are named telomeres. It's a long linear stick of DNA. And it turned out that as cells divide, these telomeres, the ends, they change. As a consequence of dividing, they shorten. Our contribution there was to show that actually it's not only their absolute length that ticks away and changes over these cell divisions, but actually their three-dimensional structure. They change in shape. We focused on a particular element of these telomeres, and we showed that once the cells senesce and stop dividing, actually there's a breakdown of this component of those telomeres, of these ends. And that really provided another way of looking at how the system is triggered.
Okay, what I want to do now is take a break to go to your lab.
Mm-hmm.
So you walk us through your lab, show the kind, so we see it physically and not just talk about it, and then come back and we'll sit down and talk about the other aspect of your research, which is the multiplication of metastases and how the cell multiplies and spreads to other parts of the body. Mm-hmm. But we'll break now.